Assalamualaikum everyone. In today's video, we'll be discussing chapter 4 of O-levels biology, which is biological molecules and food testing. So this is the smallest, or you can say the shortest chapter of O-levels biology. The code for O-levels biology is 5090. So let's get started. First part of this chapter is biological molecules. So there are four biological molecules that are included in our syllabus. First is carbohydrates, second is fats, proteins, and DNA. Okay, let's get started with the first molecule, which is carbohydrates. So carbohydrates are made up of carbon, hydrogen, and oxygen because they are made up of carbon, hydrogen, and oxygen. So they are organic. They are called organic molecules. The simplest carbohydrate that you should know is glucose. The formula for glucose is C6H12O6. This is the formula that you need to memorize. Then, when carbohydrate molecule combine together, they form complex carbohydrates such as cellulose, starch, and glycogen. I hope you would be familiar with these names: starch, glycogen, and cellulose. They are complex carbohydrates which are formed when simple carbohydrates molecule combine together. Okay, simple carbohydrates is glucose, which is C6H12O6. Now, the second biological molecule that you need to know is lipids, or you can say fats. They are the same thing. They are also made up of carbon, hydrogen, and oxygen. That's why they are known as organic compound. The solid form for fat is, or you can say of lipid, is called fat, and the liquid one is called oil. Okay, now when fatty acids and glycerol combine together, they form lipid. When three fatty acid and one glycerol combine together, they form lipid. In P2, this structure may come and they would for in the part A of the question number one for any question, and they may ask you that which or name the above molecule. So its name is lipids. Sometimes in P1 or in P2, it may come that how the lipids are formed, like which equation for the formation of lipid is correct, which would be fatty acids plus glycerol makes lipid. Now, the third molecule is proteins. Proteins are also complex molecules and they are made up of simple molecules known as amino acids. So, these amino acids are made up of carbon, hydrogen, oxygen, but this time nitrogen is also present in them. So amino acids are made up of carbon, hydrogen, nitrogen, and oxygen. However, fats and carbohydrates are made up of only carbon, hydrogen, and oxygen. When 20 amino acids combine together, they form a protein. So there is a chain of 20 amino acids. I have drawn an example chain here. This contains 20, you can say, amino acids. They may vary in shape. Let's count it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So they are 20. Now, the fourth molecule that you need to know about is DNA. So, the full form of DNA is deoxyribonucleic acid, and DNA is a complex molecule. It is made up of simple molecules, which are nucleotides. A DNA consists of a nucleotide, a ribose sugar, a phosphate group, and a protein base. So this was all you need to know for DNA in this chapter. In further chapter, we will study DNA in detail. Now let's move towards the food testing, which is the second part of this chapter. So there are three food testing that you need to remember in O level for this chapter in O levels biology. First is for reducing sugar, second is for fats, and third is for proteins. So tests for reducing sugar. Reducing sugar are tested by the biotic solution, and the initial color of biotic solution is blue. Now I have written these tests in a written form in a detailed way. So sometimes these tests come in P4 and they ask you to write or design a test. In that question, if they ask you for the reducing sugar food testing, so you can write it easily. Firstly, you need to take 2 to 5 ml of the given sample in a test tube and add the same volume of the biotic solution to the given sample. Then you have to shake it and you have to heat it using a warm water bath. So here are two safety precautions that you need to take. And sometimes in P4 they usually come, which are you need to wear goggles and gloves while heating. Now what you have to do is next to observe the color change. If reducing sugar traces are present, so yellow color appears. And if the reducing sugar is in moderate amount, orange color appears. And if the reducing sugar is at high amount, so red color appears. For P1, you can just remember that biotic solution is used to test reducing sugar. The initial color of biotic solution is blue. When this blue color converts to into red color, it means that biotic it means that reducing sugar is present in the food molecule or the food sample. Then we have test for fats. It is tested using ethanol and ethanol is transparent. Okay. Now what you have to do is to put the given sample in a test tube and add a few drops of ethanol and shake it. If white emulsions appear, fats are present. So the thing you need to remember for the fat testing for P1 is that white emulsion appears when the ethanol is added. White emulsion appears means ethanol, or you can say the fats is present in the food. Then the third test is for protein. So proteins are tested using the butyrate solution and the initial color is blue because of copper sulfate solution. So this is an additional information that butyrate solution is made up of sodium hydroxide NaOH solution and copper sulfate CuSO4 solution. You don't need to remember this. All you need to remember is that the initial color of butyrate solution is blue. When you add some butyrate solution to the given food sample, if the color changes from blue to purple, violet, or lilac color, so proteins are present. For protein test, you need to remember that blue color changes to purple or lilac color. It means that proteins are present. So this was all about this chapter. I hope you understood it. Thank you.